Hi, you guys. I am going to walk you through some genetics terms and a few examples from monohybrid crosses specifically. So let's first go over some of the terms that were covered thus far. Right now, we have dominant versus recessive. So remember that, and these are really key words to understand in order to understand how to set up and complete Punnett squares. You're really going to have a good understanding of what these four terms mean and how to set it up. So dominant simply means capital letter. That's represented by the dominant trait is always represented by a capital letter. So let's just use B's. So capital B is dominant. Typically, we represent traits with two alleles, but when we get further in genetics, we'll talk about multiple alleles as well. But for now, we're going to represent traits using just two letters or two alleles. So dominant alleles are either going to be represented by two capital letters or one capital, one lowercase. Recessive are lowercase. Are only lowercase letters. That's the only way that we're going to represent a recessive trait is by using lowercase letters like that. So if something is dominant, it has to have at least one capital letter. That's what dominant is referring to. If a trait is recessive, it must have both letters being lowercase. Next two terms, homozygous and heterozygous. Homozygous means same, right? The prefix homo means same. So looking back at our alleles from up here, big B, and little b, both of these would be considered homozygous. This top one is considered homozygous dominant. This bottom set is homozygous recessive because they're lowercase. So homozygous dominant, that didn't really work out very well, and homozygous recessive. The only combination for a heterozygous trait is one of each, one capital, one lowercase. Homo means same, whether it's same capital or same lowercase. Heterozygous is going to mean one of each, an uppercase letter and lowercase. Moving on, excuse me as I adjust this. Moving on to a couple examples. So if we're looking at flower color in a plant, and we are given the information that pink flowers are dominant, capital P, white flowers are recessive, lowercase p. If that's the information we're given and we're asked to find phenotypes and genotypes, remember that phenotypes are the physical trait. It's what you're seeing. Genotype are the letter combinations. So phenotypes for pink, a pink flower, because pink is dominant, the phenotypes phenotypes physical is going to be pink or white, depending on what we're looking at here, what alleles. Your genotypes are going to be the letter combinations. So to get a pink flower, because pink is dominant, you would either have two uppercase letters like that is a pink flower representing, or one of each like so. The only way to get a white flower, which is recessive, is to have two lowercase p's like that. That would indicate white. So phenotype, the physical trait shown, genotype, the different letter combinations that you may get from that cross. All right, moving down to another example. So if I said brown eyes is dominant, capital B, to blue eyes. Blue eyes is recessive, lowercase b. If my scenario is that I have an individual that is homozygous dominant being crossed with a heterozygous individual, how would I set that up? So first you just break it up. So homozygous dominant. Homo meaning same, dominant meaning capital. So I must have two capital B's to represent homozygous dominant. Crossing with heterozygous. Hetero means different, right? So one of each. Big B, little b. You then complete your Punnett square. putting one of the parents above, one along the side. Doesn't matter which spot they go. You pull from the top and the side to combine them. Big B, same over here. Big B from here and over here, down and across. Continue on filling in your Punnett square options. So these are my potential results. So for every offspring that these two individuals have, the likelihood of their children having brown eyes or blue eyes are the results of the middle. So here we look. Okay, so we have two different genotypes. 50% of the genotypes are big B, 50% of the genotypes are big B, little b. Physically, so phenotype we can say, physically, all of their children would have brown eyes. All of them, 100%. Because all of them would have at least one capital B, one capital letter, one dominant trait. Dominant in this is brown. So they would have no blue-eyed children. The only way to get blue, remember, is to have two little b's. All right, let's do one last example. Let me switch papers here. One last example. So now we're moving from eye color to hair color, brown versus blonde. Here I have brown hair is dominant, capital B, to blonde hair, lowercase b, which is recessive. So if I have a heterozygous individual, big B, little b, crossed with blonde. Blonde, the only way to get blonde is little b, because it is a recessive trait. Complete your Punnett square. Parents, one set on the top, the other set on the side. Pull it down and across. Now here are these potential offspring results. So here we have genotypes, genotype combinations, 50% of them, of the potential offspring, 50% big B, little b, the other 50% little b, little b. Remember, these are genotypes. That's the letters. Physically, phenotype, you're looking at 50% chance they would have brown hair. From these two right here, show that. And 50% would have blonde hair, represented here. So every offspring that these two individuals have, every single offspring has a 50-50 chance of having brown hair or blonde hair. So they can have, let's say they have two kids. It doesn't automatically mean that one is going to have brown hair or one is going to have blonde hair. That ratio resets for every individual, every offspring that's produced. The chance is 50% brown, 50% blonde. Hopefully that helps kind of clarify some of those terms and show you some practice with that using Punnett squares. Oh, and I'm sorry, you didn't see that last part maybe. There you go. All right, let us know if you have any other questions. Thanks.